Good morning, students. Today again I am here for the next lecture, that is urine formation part 2, tubular reabsorption and tubular secretion. The first part was glomerular filtration. Now the second is tubular reabsorption and tubular secretion. So let's just start with this new topic and ultimate steps of the urine formation. Tubular reabsorption is the process by which water and other substances are transported from renal tubules back to the blood. The reabsorbed, that is 99% substances, move into the interstitial fluid of renal medulla, move into the blood in peritubular capillaries, and then this tubular reabsorption is known as selective reabsorption because only the essential substances have been reabsorbed and move back to the blood. Reabsorption occurs by two processes. It may be the active reabsorption or it may be the passive. Active reabsorption and the molecules moves against the electrochemical gradients. It needs liberation of energy, which is derived from ATP molecules. Substances reabsorbed actively from renal tubules are sodium, calcium, potassium, phosphates, sulfates, bicarbonates, glucose, amino acids, ascorbic acid, uric acid, and ketone bodies. Passive reabsorption, movement of molecules along the electrochemical, that is downhill gradient. It doesn't need energy. Substances reabsorbed passively are chlorides, urea, and water molecules. Tubular reabsorption in tubular lumen in the peritubular cavities occur by two routes. It may pass through the cell or it may pass through the intracellular spaces, and these methods are known as transcellular and the paracellular. Reabsorption of the substances occurs in almost all the segments of tubular portion of nephron. PCT, in this area 88% is being reabsorbed. The brush border of the epithelial cells of PCT, they provide a larger area for the reabsorption. In the loop of Henle, sodium and chloride molecules are being reabsorbed. However, the reabsorption in DCT are of the molecules of sodium, calcium, bicarbonate, and waters. Regulation of tubular reabsorption, that how this process is being regulated. To regulate the reabsorption in tubular renal tubules, there are three factors which work behind it. That how much the quantity of the sodium to be reabsorbed, how much to be secreted. So there are the three factors. First is tubular balance. It is the balance between the filtration and reabsorption of the solutes and water in the kidney. And result is the constant reabsorption of the solute, particularly the sodium and water from renal tubules. Another factor is the hormone. Aldosterone, angiotensin second, ADH, parathormone. These are the hormones which increase the rate of absorption. Aldosterone is responsible for sodium, angiotensin also for the sodium in ascending and distal tubules. ADH, it plays an important role in the reabsorption of the water molecules. Because the filter membranes, they don't allow the water molecules to enter inside. And this is the hormone, antidiuretic hormone, which help them to enter through the filter membrane back. Parathormone, it is also responsible for the reabsorption of the calcium, magnesium, and hydrogen. I have shown these arrows. All these hormones, they increase the rate of reabsorption. These three hormones, that is atrial neurotrophic factors, brain neurotrophic, and calcitonin, they decrease the rate of reabsorption. If the amount is higher, then they decrease it. Sodium transport, sodium reabsorption is being regulated by atrial neurotrophic and brain neurotrophic factors, whereas the reabsorption of phosphate is being controlled by calcitonin. Activation of sympathetic nervous system increases the tubular reabsorption, particularly of sodium molecules. The stimulating secretion of renin from juxtaglomerular cells It also regulates the sodium reabsorption. The renin attacks on the proenzyme that is angiotensin tensinogen, which is converted in the presence of ACH in angiotensin first and then second, and which increase the sodium reabsorption rate. There are some thresholds in the plasma concentration at which the substances being appear in urine. We used to call it renal threshold and designated by Tm value. The each and every substance have a own threshold values. For example, the sugar in our blood plasma, it have 180 threshold value, that is milligram per dl. But if the value is increased about it, then it appears in the urine. And if the value is within the range, so whole the glucose is being reabsorbed and back moved to the blood capillaries. Reabsorption of sodium, it passed through the three routes, that is tubules into the tubular epithelial cells, then cells to interstitial fluids and interstitial fluids to blood. The route of move back to the blood capillaries. In exchange for hydrogen ion by antiport sodium counter proteins in PCT, and along with other substances like glucose and amino acids by symport sodium co-transport proteins. These antiport and symports, these are the plasma proteins. Antiports means they move in opposite directions. Symport they move in the same directions. And these are the plasma proteins which are found in our plasma membrane. They are responsible for the exchange of the ions. But this diagram you can understand very well. There is a movement that is known as uniporter in one direction only. Hydrogen ions, they move in a one direction. The hydrogen ions or the sodium, they in a one direction they move. That is known as uniporter. Symporters, when the molecules same means same, they move in the same direction through the plasma proteins located on our plasma membrane. Whereas the antiporters proteins, molecules moves in opposite direction. One molecule comes out and other moves inside the cytoplasm. So this is the symporter and antiporter mechanism of exchange of sodium ions. Now, how the being sodium ions being transported from interstitial fluids to the blood? Definitely, it will work again with the concentration gradient, and DCT tubules. The reabsorption is stimulated by aldosterone hormone. As I have told, it plays an important role. Aldosterone hormone. Aldosterone is being secreted by endocrine gland, that is adrenal cortex. Aldosterone also combines with the vasopressins also. Reabsorption of water from DCT tubules and collecting tubules involves facultative water reabsorption. What is facultative reabsorption? Facultative reabsorption, I have told you earlier, it is being enforced by an antidiuretic hormone, that is ADH, because the water molecules are not permeable to DCT and CT, and ADH is the hormone which is responsible to make it permeable through these tubules. And this ADH, it combines with the plasma proteins. We used to call them aquaporins. I have told you 5 types of aquaporins have been identified. It also combines with the vasopressin receptors in the tubule epithelial membrane and activates adenyl cyclase. And this adenyl cyclase to form the cyclic AMP. The cyclic AMP activates the aquaporins which increase the water reabsorption. These are the special proteins located in our the plasma membrane. Aquaporins are the membrane proteins, as I have told you, and they work as a channel for the reabsorption. Amino acids are completely reabsorbed in PCT, that is proximal converted tubule, through the sodium pump mechanism. Reabsorption of glucose, it is very important. First thing, that total glucose molecules are being reabsorbed back in our blood capillaries through the PCT tubules. And this reabsorption occurs through the special kind of proteins located in our plasma proteins. We used to call them sodium dependent glucose co-transporter proteins or the carrier proteins called glucose transporter 2. In a short, we designate them as GLT. There are 14 types of helix has been identified in human body in the plasma membrane. Similarly, the 14 types of genomes have been reported in the plasma membrane of glucose transporter. These are the carrier proteins which help to transport the glucose molecules through the plasma membranes. Bicarbonate is reabsorbed actively in PCT in the form of carbon dioxide. Bicarbonate is mostly present as sodium bicarbonate in the filtrate. And all bicarbonate ions are reabsorbed in PCT, loop of analyze in DCT. 80% of it reabsorbed in PCT, 15% in loop of analyze, and only 5% of glucose reabsorbed back in distal convoluted tubule. Now here are the special kind of proteins, that is SGLT and GLT, gluts and slits. SGLT, these are the special kind of proteins which transport glucose and galactose molecules located in the plasma membrane. This is the cell. Whereas the GLTs, they are again a different type of proteins which transport fructose molecules or the glucose molecules inside the cell. And from the cell, they transport to the blood capillaries. As I told you, total glucose is being reabsorbed back to the blood capillaries from the lumen. And it has a particular threshold area, threshold value. If the threshold value is higher than 180, then it's secreted out in the urine. Otherwise, the complete glucose molecules are being reabsorbed back, whether they are in the form of fructose, galactose, or glucose, whatsoever kind of sugar is available in our blood. Now again, by this diagram you can understand this is the cell of proximal convoluted tubule and this is the lumen and this is the interstitial space. So what happened in a lumen? We have the carbon dioxide molecules, and this carbon dioxide molecules comes from the sodium carbonic acid, break down into the CO2 and H ions. These H ions and it transported inside the cell membrane along with the calcium ions or the chloride ions, and again it forms the carbonic acids, and this carbonic acid again break down into the carbon dioxide and H ions. These are the proteins which are working in a antipode mechanism. The hydrogen ions are coming back and sodium ions are going inside. This method is known as antipode. This one is again antipode. But here is to be noted that if the potassium and sodium ions are being antipoded, they involve energy in the form of ATP molecules. 3 molecules of sodium comes out in the interstitial fluid, whereas the 2 molecules go back inside the cell. And they involve protein molecules. They involve energy in the form of ATP molecules. Similarly, here you can understand the water molecules, they pass through the interstitial cells spaces, whereas the sodium ions, they pass through this carrier proteins, chloride ions, carrier proteins, sodium ions. So this is the sympode and antipode mechanism. Antipode, one is going inside and another is coming back. One is going inside and another is coming back. So sympode mechanism, that is both are through the molecules coming inside the cell, that is the sympode. Similarly, the amino acids are being reabsorbed against the concentration gradients. Now through this diagram also you can understand another in detail. Proximal, this is the cell of proximal convoluted tubule, this is the interstitial space and blood capillaries, and this is the lumen. Here you can see all the hydrogen ions are coming outside, coming up, except the sodium, calcium ions, chloride ions, glucose ions, amino acids, phosphates. They are moving inside the cell membrane through the carrier proteins with the help of sodium co-transporter pump, or we can call it sodium pump. They move through the sodium pump inside, and this method, both are moving inside. This method is known as symport. Antiport, one is going inside. Antiport occurs, this method occurs in the interstitial fluids. In the interstitial fluids, from cell to sodium ions are moving out and potassiums are coming up inside the cell. 3 molecules of the sodium goes out and 2 molecules of the potassium comes in. So this is the method that how the sodium and potassium pump activates in our body in the process of reabsorption. Now the total summary of the urine formation, as I have told you in the first lecture, that the first step was the glomerular filtration, second is reabsorption and secretion, and some unwanted substances are being secreted in the filtrate, and few are reabsorbed, which are very essential for our body. Now what molecules are being secreted? Just have a look. Potassium is secreted actively by sodium potassium pump in PCT, DCT, and collecting tubules. Ammonia is also secreted in PCT tubule. Hydrogen ions are secreted in PCT and DCT. Maximum hydrogen ion secretion occurs in PCT, whereas the urea only secreted in loop of Henle's. And by all this process, the final formation of the urine occurs in our nephrons. By the process of glomerular filtration, selective reabsorption, and tubular secretion. And this urine formation is very concentrated and it moves forward, then it is being diluted also. So my next lecture will be on countercurrent theory, that is dilution of urine molecules. Thank you.